Welcome to a video to look at some of the aspects of genetic engineering and biotechnology. I may split this up into several smaller videos, but let's see what we can get through. Hopefully you should have an understanding of theoretical genetics, a little bit about meiosis and genes and transcription and translation, how a lot of our traits are controlled by DNA organized into genes. Okay, so first thing you should start thinking about are anything that's interesting to you in the world of biotechnology. I mean, this kind of stuff comes out in the news all the time. There's a lot of really freaky stuff that you never thought would be possible, but is now possible, or things that seem impossible now but could be possible in the future with regards to biotechnology and what we can actually do with our understanding of genes and DNA. So think about all of that. Here's a few silly questions. What four bases are found in DNA? You should know it's A, T, C, and G. Is Anna Anderson the real Anastasia? If you're unsure about this, check this out and do a Google search for these names and see what you can find out. Where else can DNA be found besides the nucleus? Mitochondria. Mitochondrial DNA actually is passed down from our mothers, actually, and can play a role in biotechnology. If some guy showed up in class claiming to be me, how could you prove his identity besides looking at his face and determining if he's good looking or not? There are other ways that you could figure it out. And finally, a man and his son get into a car accident. The father dies and the son gets sent to the hospital. At the hospital, the doctor says, "I cannot operate on this boy because he is my son." Can you explain that? Try to figure that out. It's a pretty clever answer in the end too, and it should tell you a thing or two about how you view the world. Alright, CSI, Bones, Crossing Jordan, NCIS. I don't know if Crossing Jordan is still airing, but a whole bunch of these shows are based on investigating crime scenes. That's what's really, really cool about this. Anybody who wants to be a crime scene investigator or wants to be one of these people in one of these shows here, the principles are all based off of biology and science and using our understanding of biology to help us identify criminals, or things like helping us figuring out who is the baby's real daddy, or paternity tests, which can be good news or can be very sad as well too. So DNA profiling uses a technique called gel electrophoresis, which we're going to go into in some more detail. But basically what DNA profiling means is you're looking at a sample of DNA and trying to use it and compare it with other sources to see if there's a match, or you see if there's a match. If there's a match, that could be a paternity test. It could be to identify a killer or to help solve other types of crimes as well. Who's my daddy? Paternity suits, the killer or the rapist, if they're comparing various types of DNA samples for crime investigations, and also for identifying dead people. So one of the previous questions that came up about who is the real Anastasia, Anna Anderson was one of the most famous issues that came out from there, and some biotechnology helped to answer that question. They could actually compare some DNA from people who had been dead for a long time and actually check to see if she could possibly be a direct relative from there. So it's really amazing what we can do. Some of these shows stretch the truth a little bit, but it's not all like Star Trek stuff. So is Star Trek based on some truth? Maybe. But these shows are definitely pretty scientifically accurate, and you'll find out about some very interesting things by watching some of these shows as well too. So if you need any time wasters, go watch some of these shows and then get back to studying. Okay, so DNA, how do we actually analyze this? So there's a process here called PCR, which stands for the polymerase chain reaction, and a PCR machine is pretty expensive, but what a PCR machine does is it amplifies small quantities of nucleic acid. So if you find any samples of DNA, so usually before someone is able to do any kind of test, to test the actual presence of the DNA and match it with a sample, they usually have to amplify it or make more copies of it. And so PCR, all you need to know is it's called the polymerase chain reaction, and it's a process that we use to amplify amounts of DNA. So if I find someone a crime scene in a blood sample, semen sample, hair samples, or anything like that, one of the first things that's done in a lab is it's actually amplified. You don't have to know details about the process, but it helps us to actually make larger amounts or copies of the DNA, because you need more copies in order to do some of the other more sophisticated analyses like GL electrophoresis. So the purpose is to make many sequences of a particular DNA sequence very rapidly. And it's just like an artificial version of DNA replication. Quick question, should be easy. How is the polymerase chain reaction, or PCR, used? Pause the video. Good! Congratulations. For each of these others, see if you can figure out what the right answer is. If this is just your first video about biotechnology, you may have some difficulty answering some of these questions, but see if you can figure those out, or come back to it if you have a chance, or take a picture with your phone and then revisit it next time. Okay, so this is called gel electrophoresis. It's a very fun technique to do and to analyze as well too. So what we're looking at here is a gel which has the consistency of, all I can think of is Japanese food. Something called konnyaku. It's kind of like a rubbery jello, kind of hard jello. So imagine hard jello, okay? And you touch this stuff and it's clear, you can see through it, and under the correct lighting, or UV light, particular bands can actually light up as if you've added something that makes it glow under UV light. And what we're looking at here are bands of DNA, and we have to look at this in more detail to understand, because each one of these lines is not one chunk of DNA. Each one of these lines is a pile of a bunch of strands of DNA that are all the same lengths. So there will be various activities you might try out in class that could help you visualize what's going on here to do this entire process. But what it does is it separates DNA. So if I take a big chunk of DNA that I found in some kind of crime scene, what I'll do is I can cut it up with a whole bunch of miniature scissor molecules called restriction enzymes. And when I cut them up and drop them into a little well like this and turn on some electricity, what will happen is those chunks of DNA will actually get separated by their size. The ones that move further along are going to be smaller. The ones that don't move so far are going to be larger. So they're being separated according to their charge and size. And we're going to look at a short animation from the DNA Learning Center. Do a Google search online and you can download their little animations, and I'm going to show you one shortly. So it's a process used to separate fragmented pieces of DNA, and that DNA is separated according to the charge and size. So I'm going to switch over right now to take a look at this animation. I'm not going to read everything to you here, but please check it out at that website. But I'm just going to use the diagram. I'm just going to talk over it really quickly to help you understand what's going on. So there's that gel. We call it an agarose gel, and it has these wells that are cut into it. The wells don't go all the way through, because if they did, then if I put something into the wells, then it would just drop all the way onto the bottom. But so these wells are actually cut about halfway down deep into this actual gel right here. And the gel is placed inside some liquid that allows electricity to pass through, and it's hooked up to a power supply right here that we're going to turn on in a second. But first, we need to put our DNA that we've actually amplified, and there's DNA. Now the DNA that we're putting in here has already been chopped up, using specific little miniature scissors. I'm gonna keep saying that they're called restriction enzymes. And once they get chopped up into little pieces, we're actually going to put them into the well and turn on the power supply. Now, it's very important here, the negative side and the positive side. The positive side is always towards the far end, because if you look at the structure of a nucleotide, nucleotides are what make up DNA, the phosphate groups are actually negatively charged. So DNA has a negative charge overall. So if I turn on this power supply here, the DNA is going to move towards the positive end. Let's check it out. So we add this other blue colored dye so we can see how far it's going. But this is what's happening. Those chunks of DNA, the smaller chunks, the gel is kind of like it's a matrix here with all kinds of these proteins that have made chains. And so if you imagine crawling through here, it'd be a bit of a mess, hard to get through. You got to push your way through. If you are small, you can squeeze through a lot faster. If you're larger, you don't go through as as fast. And so that's a great way to separate out these DNA fragments by their various sizes. So continuing on, now we've added a blue dye just to help us visualize. The blue dye is just to show us with the DNA has come all the way to the end. If we leave this on forever, then all the fragments are actually going to drop into the liquid and we're not going to get a good picture taken. So now that everything's all done, we've added a dye in there called ethidium bromide. And ethidium bromide is not that blue tracking dye. So there's two different dyes. One is the blue tracking dye and the other is ethidium bromide. And ethidium bromide will glow when illuminated under ultraviolet light. And then here you can see that these fragments are moving. And so I can conclude that these fragments here this is a pile, a clump of a whole bunch of DNA fragments that are all the same size. Hence they move together. And the more fragments that are there, the darker this little stain will actually be. Okay? The less fragments there are, the lighter the stain will be. So I can conclude this was the negative, this was the positive side, and then these DNA fragments that were cut. These names up here are representing the different types of molecular scissors called restriction enzymes that we're going to see a little bit later in subsequent videos as well too. But that is one of the main functions. Then we can take a picture of the entire thing and analyze it. So let's give one quick example of how we could analyze some of these things. There's also you could do a search for gel electrophoresis virtual labs online, and you can try to do this process virtually. It's pretty fun as well too. So take a look. A few things to remember, but what I really want to do is get to a little sample question. DNA is what kind of charge? Negative, because of what? The phosphate groups of the nucleotides. It will move towards the positive end. The smaller the fragments, the farther it will move. Our DNA fingerprints, all DNA fingerprints are unique, true or false? And the answer should be true, but of course you can have twins as well too. What are the bands actually showing? They're showing clumps of DNA of different sizes. And so an application question really quickly, something like this. You can use these kinds of images to determine who the real parents are of a child, or who the killer is if you're matching a blood sample with another blood sample. In this case, this one is quite interesting. If 1 and 3 are the parents and number 2 is the child, what you end up with, and this is very clever how this works, if all the samples are cut with the same type of molecular miniature scissors called restriction enzymes, then every band that you find in a child will match up with a band either in one parent or the other. So in this case here, every band that shows up on this child here will actually match up with a band from one of the parents. So for example, if this band on this child, if this parent number 3 didn't have this band, then none of these parents actually have this band. That tells me that one of these parents is not actually the real parent. And now you can even do over-the-counter DNA paternity tests by buying a little kit for a few bucks, and you do a little sample and you send it. The actual analysis might be a little bit more expensive, maybe like $80 to $100, but you can actually do this on your own if you suspect that a parent is not who they say they are. So that's kind of scary. I don't know about the statistics if you want to find out more about that stuff. But there's gel electrophoresis, some of its applications, and a little bit about some of the introduction technologies to biotechnology. Sorry that video was a little bit long, but watch it in 2 parts. Yes.